मैं राजवीर सिंह शेरान एसोसिएट प्रोफेसर इन केमिस्ट्री SRR मोरका गवर्नमेंट PG कॉलेज झुंझुनू. First of all, good afternoon to all the students of MSc final. Yesterday we discussed the enzyme models of crown ether and another enzyme models. Today we will discuss the next enzyme model, calixarenes. So first of all, we define what is calixarenes. Calixarenes are cavity-shaped cyclic molecules made up of benzene units. They were named by SO Govt because of their resemblance with calyx type in Greek language was cup-shaped. Greek was calyx means cup-shaped. Functionalized calixarenes can act as new type of catalyst, ligand and host molecules. In fact, calixarenes are group of phenolic macrocycles obtained by condensation of para-alkyl phenols. For example, para-tertiary butyl phenol with formaldehyde and containing repeating units as in a tetramer K and L. This is designated as para-alkyl calyx 4 arenes. So calixarenes from 4 to 14 subunits are known. Hydrophilic phenolic groups are at the bottom of the cup and form pedestal or legs of calyx as shown in figure. So this is cup shaped and four hydrophilic groups, hydroxyl group ions are attached at the bottom of the cup shaped calixarenes. So we can discuss the cavitant groups. Calixarenes are examples of which act as molecule vessels without bottom, can hold ions as well as neutral guest. Hydrophobic cavity of calixarene form sigma pi complex with sterically compatible neutral molecules like toluene, xylene, pyridine and anisols, etc. Phenolic group of calixarenes can be derivatized, for example acylated, alkylated, etc. and new array of functional groups, for example ether, ester, thiamides, amides, etc. can develop primary binding sites. For instance, tertiary butyl group can be expelled and replaced by hydrophilic groups like sulfonic acid group, changing receptor property of calixarenes. Chemically modified calixarenes are good receptors both for neutral molecules and cations, including some transition metal ions. Phenolic groups of calixarene get deprotonated in alkaline medium and are capable of binding with cations complexes thus formed, so ionophoric properties and transport the ions like liquid membranes. Octamers, calixarenes show fastest rate of transport with cationic selectivity of the order Cgm plus or greater than rubidium plus, greater than potassium, sodium and lithium. Lanthanide form complexes in basic medium with tertiary butyl calyx 8 arenes which gets dissolved in organic solvents, leading to efficient method of extraction and purification. There are also evidence for metallic tunneling through calyx 4 arenes cavities. PMR and proton magnetic resonance at low temperature -50 degree to -85 degree has shown that Silver plus ions alternate intramolecularly between two binding sites through pi basic hole of the 1,3 alternate calyx 4 arrays conformer. This is the first example of silver plus ion tunneling across aromatic cavity not only of calyx ring but also some other ion channels, metal inclusion of fullerenes, interaction of metal cavities into graphites, etc. Next enzyme model is ionophores. Some polyether form strong complex with alkali metal ions selectively, which being soluble in organic medium behave like ionophores. Actually, ionophores are polyether, polyester, polyamide usually arranged in macrocycles and have capability to bind with metal ions and transport them through biological and artificial membranes. Some examples of each polyethers are as follows, as shown in the following diagrams. Cryptands. So we can say naturally occurring ionophores are mainly found in microorganisms and a number of them are used as antibiotics. They interfere with permeability of bacterial membranes by coordinating with metal ions. Some examples of naturally occurring ionophores are discussed below. Non-actin, it is an antibiotic which works by transporting sodium ions into bacteria till increasing osmotic pressure reaches the point of rupture of cells. Valinomycin, it complex selectively with potassium ions where permeability of cell membrane for potassium plus ion is offset. Then antanomide, it is a D-capetide containing L amino acid only and has a binding cavity of different geometry in comparison with valinomycin. It also shows preference for sodium plus ion over potassium plus ion. And nigericin, they causes reversible ring closure upon binding with metallic ions. They show effect opposite to valinomycin. Like this, antamidin and peptide ionophore, proline, alanine, phenylalanine, then proline, phenylalanine means cyclic type जो antamides are formed. Then micelles. Micelles surfactants are amphilic molecules as they have both pronounced hydrophobic as well as hydrophilic characteristics. For instance, a detergent is an excellent example of cleaning action involving this principle. In solution, low molecular weight electrolytes forms ion pair with current counter ions. Upon increasing monomeric concentration, clusters of low molecular weight electrolytes are formed. These are large in size. Aggregates are known as micelles. Micellization of monomeric surfactants occurs when surfactant concentration exceeds critical micelle concentration means CMC. Generally, critical micelle concentration ranges from 10 raised to power minus 2 to 10 raised to power minus 4 molar, and conductivity of solution charges sharply above this concentration. Now we discuss the structure of micelles. Micelles are often spherical. In polar medium like water, the hydrophobic hydrocarbon chain of surfactant is oriented inwards in the sphere, whereas polar or ionic heads are directed on the surface of the sphere and are surrounded by aqueous solution containing counter ions. Proximity of hydrophobic group in water leads to favorable entropy change due to liberation of water molecules from the aqueous polar interface, and hydrophobic groups get considerable freedom of movement in micelles. This increase in entropy results in favorable free energy change in micellization. Micellization can be explained by taking example of surfactant dodecyl trimethyl ammonium bromide, which creates positively charged surface made up of cationic heads. Bromide ion has bromide ion come in vicinity of quaternary nitrogen because of Coulombic attraction. This negative region form eastern layer. Very little water is present in the interior of micelles, which contains hydrocarbon part of surfactant. Difference of polarity between interior and surface of micelles make it resemble global protein. The active site of enzymes are also non-polar, although the enzymes as a whole are water soluble. Thus, micelle formation may also be involved in its action. Organic compounds, particularly non-polar organic compounds, can be absorbed into or in the micelles. Thus, their solubility relative to water is increased, that result in change in their chemical reactivity. It is micelle, rather the surfactant, that result in alternation in rate of chemical reaction in aqueous solution of surfactants. Proper choice of surfactant leads to 5 to 1000 times increase in the rate of reaction. Depending upon the type of micelles, concentration H plus ion or OH ions stain layer is increased, which is responsible for increased rate. Nucleophiles or other group also catalyze the reaction. However, interaction between Govt-Chapman layer and diffused double and micelle is weak and results in gradual in ion gradients. It is worth mentioning that Govt-Chapman double layer extends several hundred angstrom from micelle surface. Reaction rate accelerates can be formed cationic, anionic as well as non-anionic micelles. Some examples are illustrated as below. For example, A. Bis 2,4-dinitrophenyl phosphate is rapidly hydrolyzed at pH 8 by cationic micelles. Then deuterion molecules. Deuterionic micelles are relatively ineffective in catalyst but are responsible for the rate of enhancement of following type of decarboxylation. As shown in the reaction, CO2 is removed, CHCN and H plus CH2Cl. Sodium dodecyl sulfate SDS surfactant contain 50 to 100 molecules in sphere. There is 50 to 100 millivolt potential difference between bulk and micellar phases. Electrostatic and hydrophobic interaction are main factors for maintaining stability of micelles. This surfactant is responsible for denaturation of proteins in which similar forces are present in tertiary structure. Then cyanide ion has react with N-alkyl pyridine salts. Its rate is considerably enhanced by cationic surfactants. Longer the alkyl chain, faster will be the rate of reaction. This indicates that hydrophobic binding is an important reason for the increased rate of substitution in the reaction. Then synthetic enzymes or synzymes. The rigid macromolecules having catalytic imidazole group and hydrophobic groups similar to micelles are closer in structure to enzyme like synthetic polymers and are also known as synthetic enzymes or C enzymes. The Their reactivity is same order as that of enzyme. Then synthetic strategy. Amino acid residue present in all the enzymes can be divided into three categories. Number one, active center residues. Number two, other residues which are required for enzyme conformation. Number three, non-essential residues which are present in outer periphery. Then experimentally we have revealed that slight change in either the category A or B residue can be considerably alter or even eliminate the catalytic activity of the enzymes. Thus, initial attempts to synthesize enzymes focus on the alternation of only one residue or a limited number of residues at a time, most likely an active center residue. The strategy used to do such alternation may be delineated as follows. In the given reaction, amino acid and then X polymer, it combines, gives the product. Then this again reacts, and this coupling formation is formed as shown in the diagram. Scheme for solid phase peptide synthesis. X-ray diffraction, in addition to information obtained from other traditional chemical sources, is used to identify active center residues. Such type of reactions are generally seen. We can see hypothetical peptide containing three large constant fragments and two small variable fragments. These enzymes can be examined by traditional chemical method for catalytic activity, thermal stability, and substrate specificity. Use of large constant fragments, which leads to synthetic enzyme possessing almost perfect homology when compared to parent enzyme, hopefully minimize potentially serious problems related to different modes of polypeptides chain folding. Any change in folding pattern from parent enzyme may lead to change or disappearance of active sites. Success in synthesis of enzymes depends upon a knowledge of a primary sequence of parent enzyme and three-dimensional structure of enzyme in presence of transition state analogous what residues are essentially for catalytic activity and substrate binding as shown in the following compounds A and B dash. Now what are the applications? So by the preparation of enzymes, it is possible to alter the catalytic activity, substrate binding, pH behavior, susceptibility to inhibition, etc. Enzyme immobilization on solid support may be facilitated and work may be done to elucidate and confirm enzyme action mechanism. Enzymes may have reduced tendency to enzymes to denature, may have the entirely new catalytic function and enhance the solubility in non-polar solvents. In fact, successful commercialization of enzymes depends upon our ability to chemically modify natural enzymes. Alternation in a single residue is different to bring conformation change in the molecule. For instance, in hemoglobin, it accounts for observed metabolic differences in characteristics of mutant hemoglobin. So, it has been found that the soluble blocked amino acids with amino acid coupled to non-cross-linked polystyrene resin reacts 100 times faster as compared to cross-linked polystyrenes resins and follows second-order kinetics. Para-nitrophenyl active esters and dicyclohexyl carbonamide DCC method also gives rate increase of the same order. So it has been found that beta pyrolyl 3 alanine B dash when substituted for histidine in angiostensin H plus also yields a peptide with significant pharmacology activity in the laboratory animals. Thank you.